Today on the Arabidopsis Research Roundup, I'm joined by Professor Anna Amtmann from University of Glasgow and the lead author on a paper, Giorgio Perella. And the title of this paper, new paper which is published in Plant Physiology, is the Histone Deacetylase Complex. One protein of Arabidopsis thaliana has the capacity to interact with multiple proteins, including histone 3 binding proteins and histone 1 variants. So thank you very much, Anna and Giorgio, for joining us today. And so could you give us a brief summary of the paper, please? That'd be great.
Yes, hello. So I'm Anna Amtmann, and I'm here with Giorgio Perella, the first author of the paper. And this was a really exciting story. So histone deacetylation is a process by which plants can modify the chromatin, and these modifications have important implications for the transcription of genes. So the enzymes that actually deacetylate histones, in particular we are talking here histone 3 and histone 4, don't work alone. They work in protein complexes, and a couple of years ago we actually identified a new protein which we called Histone Deacetylase Complex protein 1. There is actually only one, but one, which is a component of these multi-protein complexes. And the exciting thing about HTC1 was that it really has a quantitative effect on the actual activity of these histone deacetylases. So if you overexpress histone deacetylase complex 1, so HTC1, if you overexpress it, you get a range of phenotypes related to flowering, germination, and so on. Mm-hmm. And when you knock it out, you get the same phenotypes in the opposite direction. So meaning that you have actually a quantitative effect both if you have more or less of this particular protein. And this is quite interesting if you think about it, because in order to have an effect within a multi-protein complex, in a way, you need to be what we would call the rate-limiting component to have really effects in either way. Yeah. So this is what made HTC1 really exciting in the past, and we have actually collaborated with the company Bayer here because the downstream effects, the phenotypes of HTC1 include growth, for example, particularly also under stress conditions. So this has potential implications for crop performance and so on, and Bayer has since actually taken that gene into their crop improvement program. Okay. So HTC1 is an important gene.
Right, okay.
Now, in this paper in Plant Physiology is now a follow-up study, and Giorgio will quickly describe what he did.
Okay, cool.
So in this paper that has been recently published in Plant Physiology, what we look at is the capacity of HTC1 to bind with the other complex partners. Okay. Because based on previous studies where we show that HTC1 is able to bind to the catalytic subunit based on the acetylase, we then decide to follow that up and look what was the possible interaction between HTC1 and the other putative complex partners in Arabidopsis. So firstly, what we did was to pull out the homologs from the yeast complex to Arabidopsis. Yep. And look at interaction between HTC1 with the other complex partners. And what we find out is that HTC1 is able not only to bind to the acetylase, but is also able to bind to the other complex partners, especially on the histone binding side. Okay. So we find interactions between HTC1 and histone binding proteins, such as SHL1 and ING2. And at the same time, we find binding between HTC1 and the core factors such as SAP18. But interestingly enough, we do not find any binding between HTC1 and the core repressors, which are part of the acetylation complex, such as SNL2 and SNL3. Okay. Then on the other hand, the other interesting finding on this paper is that HTC1 has also the capacity to bind with the histone 1 protein. Okay. And that's a very new finding because so far only a few papers have shown the ability of histone 1 to bind with other proteins.
Okay, so the relevance of that is that histone 1 is thought to, because it's not modified in the same way as H3 or H4. So, I think the function of histone 1 is thought to be more of a linker between the octamer. So, what do you think the functional relevance of this binding of HTC1 and the H1 might be?
so the exciting thing is that histone 1, as you say, is so-called the linker histone. So it actually is at the fringe in a way. So the nucleosomes, which are wrapping around the DNA, are made up from H2, 3, and 4 histone proteins. And the histone 1 sits at the periphery and sits on the linker DNA between those nucleosomes. And, I mean, what is very well known is that histone 1 is really important, actually, for chromatin structure as such. But until now, its involvement in sort of phenotypes and physiological responses and transcriptional responses was not really well understood.
Yeah, okay.
And lately, particularly also in the animal field, there is actually increasing interest to seeing what is H1 actually doing other than just sitting there and, in strengthening the chromatin. And one very particularly interesting study has come out a couple of years ago by the Daniel Silberman lab, where they actually showed that in order to get access to the histones in the core nucleosomes, it may well be that you need to remove histone 1. So there are basically two ways of potentially including histone 1 into the histone deacetylation story. One possibility is that HDC1 actually makes a link between the proteins of the histone deacetylation complex to the histone 1, and that simply strengthens the whole complex as such. So that actually increases the stability of the protein by also, and even leads to further chromatin compaction by actually pulling in a way also the histone, the linker, right? So basically further compacting by making a link to the H1. The other possibility is exactly the opposite, that you actually have to remove H1 in order to get access to H3 and H4 with your deacetylation enzyme.
Okay, I see.
And we are currently, of course, now testing. This is now follow-on work that really needs to be done, because what you can test now, if you have double mutants between HDC1 and histone 1, whether you get the one or the other effect.
Okay, I see. And just finally, in the abstract, you talk about an ancestral form of this HDC1. So from that, can we infer that in more basal plants, there's a smaller protein which is X, or a protein with different motifs? And what can that tell us about the evolutionary relationship of this, or evolutionary work of this protein? Can you say something about that, perhaps?
Yeah, so I mean, one difficulty of working with HDC1 is that if you look at the sequence across all plant species, there's actually not a single region that has any sort of predicted motif or function. So it's a completely unknown protein, which of course why it had a lot of IP and patent potential as well.
Okay.
And interestingly enough, it is actually present in all higher plants. It's present in algae. And a part of it is present in fungi. So basically, there is a certain part of this large unknown protein which is conserved in algae and fungi.
Okay.
So that's what we call the ancestral core domain. It's about a third of the protein, and then the higher plants have hugely extended the N-terminal part. So what we did is in the first approach to get some sort of structure-function relationships, we cut down the protein to this core part, which we call RHT3, because that's the name in these, so this RHT3-like part. And what we found then is that most of the interactions with the other proteins were strongly diminished. So really this N-terminal extension in higher plants was very important to provide that large interaction profile that the protein has. But certain interactions, and particularly one, for example, to histone 1, was fully maintained. Okay. And we also then looked at the phenotypes of when we complemented knockout plants with this smaller part rather than as compared to the full-length part. And we could see that smaller part could complement some of the functions, for example, involved in ADA-dependent germination and growth, but not other phenotypes such as flowering or very specific leaf development. So from that we sort of conclude that maybe the binding to histone 1 and also to one of the histone 3 binding proteins is the more ancient function, and that it's required for certain phenotypes to do with very basic physiology and growth, whereas the extension being then able to bind to many more proteins, probably also in the higher plants, probably also extended the phenotypic profile and functions in higher plants. Okay. So probably what's important to say at the very end is that if you make a structural prediction of that protein, it's actually almost completely unstructured.
Right.
So we look at it at the moment as this kind of alien gluey thingy, which sort of wobbles about, and actually these kind of intrinsically disordered proteins very often actually act as sort of hubs of multi-protein networks, because they basically have the flexibility to actually bring together something.
Okay, excellent. Well, over the coming years, we'll look forward to learning more about this alien blobby part of the protein, certainly. Okay, well, Giorgio and Anne, thank you very much for joining me today. Thanks very much.
Thanks a lot.
Thank you.
See you later. Bye.